So again, thank you for your patience, everyone. I just want to begin with an acknowledgement of the traditional owners and their custodianship of the lands on which we are currently. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. And now, without further ado, I would like to hand the floor over to Professor Rezende after I have spent 15 minutes getting technology working. And I will be monitoring the chat if there are any other technical issues.
Can I start?
Yes. Yes.
Thank you very much, Simon. Thank you very much. Just to put the visit in perspective, we're supposing Nacho is a Marie Curie fellow when we come to Australia for 3 weeks. And maybe a month ago, they invited me to participate in a heat wave workshop or manuscript, and which is very fortunate because part of the research that I've been doing so far. So just to put in perspective, I started as an evolutionary ecologist and physiologist for the past, 25 years, but then I became very interested on thermobiology and eventually theoretical thermobiology, if you will. So I came more from an empirical side, and eventually I slowly drift to more theoretical analysis for the past 10 to 15 years. And the idea now is to talk about what, knowledge of a thermal tolerance can tell us about what's coming as the world keeps warming. So just to put in perspective, there's this very recommendable book by Michael Anguleta, and one of the most interesting aspects when I read the book was in the introduction, when he mentioned that thermobiologists have described patterns and documented effects to the point that empirical knowledge, right, empirical data has far outside theoretical understanding. We have way too many experiments and very little theoretical understanding of what's going on. And just to put it in perspective, as a thermobiologist, if you will, there was the seminal paper by Curtis Deutsch and colleagues in 2008. And the beauty of this paper was that for the first time, they tried to get physiological knowledge. In the case, it was thermal tolerance limits, and overlap it against predictions of what might happen with climate change. And they showed this inverse pattern where climatologists would say that climate change or global warming would be more extremes near the poles at high latitudes. But Deutsch and colleagues, they showed that actually because of the thermal tolerance of living organisms nowadays, the equator would be more impacted. So let me see if the laser works. No, it doesn't, never mind. But essentially, that the population, the communities that would be more threatened about this, smaller change in temperatures would be equatorial ones. And you can see that in this prediction here. You can see that those animals or communities and organisms that are falling underneath what would be the predicted impact are actually tropical ones, even though climatologists would say that the highest increase in temperatures would happen near the poles. So suddenly, for many people, myself included, it was the first time that we realized that physiological knowledge could be used for predictive purposes. And not only that, it would invert many times what we might expect based on climatology data or climatological forecasts. Now, the problem with that, and that's when I started working on this, is that even though Curtis and colleagues, they did this very nice work, there's some problems with the approximation. And especially the idea is that when we work with what people might call T-crit or CT max, depending on the terminology, but essentially a given temperature where animals actually die, you're working with an implicit assumption that you have a threshold model, that essentially organisms above that temperature will die, but below that temperature they should survive. And the problem, that the reason why I started working in this field was because we wanted to measure thermal limits in Drosophila. And when you start going into the literature, the thermal limits would vary dramatically depending on the protocol. So people would would put these animals in the water bath, and they would increase the temperature, and depending on the heating rates and depending on the starting temperature, whatever you got that would call the thermal limit that you create would vary enormously depending just on measurement protocol, which would imply that whatever you want to predict with these measurements was highly unreliable. Okay, and here I'm putting just a couple of examples. For instance, in here, the sea surface temperature, you can see that it varies between 0 and 30, but in here you can see that in many instances you have marine organisms, they have so-called thermal safety limits that are in the margin of 15. So essentially half of what you see geographically here. All right, or for other terrestrial organisms, you might have organisms that can tolerate, presumably they have safety margins, which is whatever they can tolerate, in contrast with what they're experiencing right now of 25 degrees. So global warming shouldn't be an issue. These animals should in principle tolerate, temperatures 25 higher from what they're perceiving right now. So there's definitely a problem. right? I mean, I mentioned that as a Brazilian that has lived in London. I know they're very different, right? And so claiming that somehow you have these organisms with this sort of safety margin, it's for predictive purpose, it's problematic. And second, from an evolutionary perspective, it doesn't make sense. How do these limits evolve? Okay, and so I started getting into this problem, if you will. And eventually what we realized is that the notion of how you measure or you estimate thermal tolerance is incorrect. It's not a threshold trait, right? It's a cumulative trait. In order to show you that, let's start with this example. Let's imagine you have a population of organisms and you put them at 30 degrees. So everybody's alive, and as time goes by, if they start dying, right? And so the survival probability curve here, the mortality starts increasing, they start, everybody dying, and it happens at a given rate with a given shape. But now if you increase the temperature from 30 to 31, the time that they take to die accelerates, right? They die faster. And the same applies for 32, 33, and eventually 35. So what you can see here is that you actually have a dependent variable, which is the probability of survival. And that depends on both the temperature we're exposing these animals and the amount of time that it takes. So in here, with this system where you have constant temperatures, you can actually separate the cumulative component, right? You're separating the intensity of a stress, which is temperature, and the exposure time, which is the accumulative part of it, okay? So with this basic idea, we came up with the notion of thermal tolerance landscapes, which essentially is this. You have this 3D model, or this 3D landscape, if you will, where probability of survival is the dependent variable, and you have on the one hand time, right? And the other hand, you have temperature. So if you're confined in this area here, where probability of survival is above zero, part of the population can tolerate, can survive, but if you're here, everybody's dead, okay? And so with this 3D landscape, of course, it's complex enough. So what you can do is you can assume a constant probability. Let's imagine LT50, so 50% of the population surviving, and you're cutting this, right, the probability of survival here at the middle, and this is the projection that you see in 2D. And this projection, it has two components. Here it's in arithmetic scale, but if you log this here, this would look like a straight line. And the straight line would have two components. It has an intercept, right? Any regular line, right? An intercept and a slope, okay? And so the intercept here is what we, unfortunately, for historical reasons, call CT max, which is the temperature that you can tolerate following a 1 minute exposure. And the reason why it's a 1 minute exposure is because T, we express it in minutes. So the moment this is log of 1, this becomes 0, this whole thing goes to 0, and this becomes an intercept. So that's your thermal tolerance following a 1 minute exposure, in this case 40. And the slope is the delta T, is how much below that limit you need to decrease in order to increase a 1 order of magnitude of the amount of time that you can resist. Okay? So just with numbers, it's easier. If you have a CT max of 40 and your Z is 2 degrees Celsius, that means that you can tolerate 40 degrees for 1 minute, 38 degrees, right? The difference, 2 degrees for 10 minutes, 36 for 100 minutes, and 34 for 1,000 minutes. That's the meaning of Z here. This 2 degree difference essentially tells you how much you have this tenfold increase in time that you can tolerate. And this is a projection, and many people call this a thermal death time curve, okay? And so in this case, we have a 50% mortality, but in reality, if you want to go and study the whole thing, you need to know how the probability of survival curves look as well, okay? So you're going to see a little bit of both here in this presentation. So in 2014, we realized that this was a general phenomenon. Now we have here the regression line fitted. You see it's log scale here. So you can actually see how empirical data, this is for Drosophila, it fits very well. I've done it for many different organisms, and the R-square is always above 0.95. Okay, so the R-squares are, in many instances close to 1. I'm going to show a few of them. Okay, this is the same graph, because you're going to see in the literature now that this relationship is plotted in many different ways, but it's the same relationship. And actually, statistically, you can do that because you have to remember that you have the effect of time and the effect of temperature. In principle, they're orthogonal, and the dependent variable is survival probability. So it's not that time depends on temperature or vice versa, right? You have a third dimension here that's the one that really matters. Okay, so you can play with that, and that I can get into more detail of why this is important. But in this case, for instance, you have that for this is Drosophila obscura, and you have an intercept of 41.4. So essentially, that's a projection here when it intercepts the x-axis in 1 minute, and you have a slope here of 3.9, but a very, very good fit. I've done the review by now. So in 2014, we had, I don't remember now, maybe 100 TDT curves and with very high R squares for many different animals. So by now, this has been shown to apply for flies, for bivalves and mollusks, for amphipods and crustaceans, many different fish species. We found a review for fish that this review was published in 1972, and we've got more than 300 TDT curves or thermal death time curves for fish. So we know it applies for animals. We know that it applies for bacteria and fungi, and that comes from the food processing literature and canning industry, because they had to sterilize, the food before they put it in cans. Otherwise, the cans would explode, literally, right? It's fermenting. And so, in this case, the biochemists in the 19th century, they were doing these relationships, and they published like these very good papers, and they were showing essentially what were thermal death time curves. And nowadays, we know, working with some people here from Canberra, that this also applies to plants for photosynthetic efficiency. Now, all right, so, I'm all here selling the case that it's not a threshold trait, it's cumulative. The question is, why does it matter, right? Why should we care? And so this table made a big deal, or it was quite important when I found these results. So essentially, we had this data set by Eric Hoffman, so he's, I think, in Monash or somewhere in Melbourne, okay? And they were putting these reviews on the variation of heat tolerance as a function of latitude, and they couldn't find anything. And the idea was that there was this, hard boundary that organisms could not, overcome, and there was no latitudinal gradient whatsoever. And so with this framework, or my framework if you want, I downloaded that data. I went to the original papers and I calculated the amount of time it took for, the protocols that they employed in the original papers, how much time was involved in these measurements to estimate CT max or heat tolerance in that case. So I ran their analysis again, first with the original measurements, the threshold data as a function of latitude. There's no latitudinal gradient whatsoever, right? So it's not significant. And the R square here is 2%. So essentially it's flat. There's nothing. But when I include now the logarithm of t, which should apply if it's really a cumulative trait, you see that it's highly significant. And by now we can explain the variation in heat tolerance by 64% of the variation observed. So this is what's been missing if you don't include t, right? If you don't take into consideration the measurement time. And by now, when I include both of them, you can see that we can now rescue, or we can really observe the latitudinal gradient is highly significant and increase now the explained variance to nearly 68%. Okay, so it matters. And in this case, just to emphasize, this remains a simplification because we were assuming a single slope for all the organisms in that analysis. We know the slopes are going to vary, okay? And in this case here, in the regression model, just the slope interval for the latitude, actually, we could calculate that CT max would decrease from the equator to higher latitudes by, an amount of 15% degrees Celsius per degree of latitude, which means more or less that from the equator up to 60 degrees north, so the difference between the equator and London would imply a difference in heat tolerance of 10 degrees. It's not a minor difference, right? It can be pretty substantial. So it matters, right? So we should be working with it. Now, 6 years later, it took 6 years because there was this problem that all that I showed you was how you can experimentally separate the effects of temperature and time, which means that you should do the analysis or the experiments employing constant temperatures. But ideally, you want to use this framework with variable temperatures because that's what matters in nature. So eventually, we were able to generalize the model, and this is how it works. These two panels here is what you observe in constant temperatures, the same thing that I showed you before, right? So you have, in this case, it's mortality. Here's the log time. So you put them in a cold temperature, not as extreme. They take some time to die from here to here. The same thing for an intermediate and then a warm temperature, right? And as I showed you, there is a relationship here that is defined by the slope. In this case, it's minus z simply because of what you use as x and y. But here you have, all right, it's z. Okay. If you do now this, you observe this relationship now as a survival probability curve as a function of time. So it's the same thing, another projection because it's 3D, right? You start with 100% survival, and then as it's cold, they take some time to die. If it's intermediate, they die faster, just like the first figure that I showed you. And if it's warm, they die even faster, right? You can now obtain the derivative of these curves. And you can compile or you can build now the expected curve as temperatures are rising from T1, from the cold temperature, to a warm temperature, T3. And what you can see is that this cumulative curve now uses the derivative obtaining constant temperatures, and you can obtain what would be the predicted result or the predicted survival curve under fluctuating conditions. And that's just calculus, right? Basically, basic calculus. And the question is, can we use this to generalize the models or to predict what happens in fluctuating temperatures? Just to put things in perspective, what this is doing, it's a very fancy way of translating high resolution temperature data. So in this case, it's Santiago from winter to summer, temperatures increasing, and you're transforming temperature data into quasi-instantaneous mortality data. We can do this calculations on a minute basis. All right, we obtain hourly values of temperature, we interpolate, within an hour, and eventually we can estimate the mortality minute per minute. And you can see in this case that as it gets warmer, for this Drosophila, I'm going to show you the results later, for this population of Drosophila that was acclimated to cold and warm, you can see how by November they start slowly dying, and then as temperatures keep increasing, you expect daily mortality to reach around 100%. I mean, the place is no longer inhabitable, if you will, right? I mean, it becomes prohibitive because of the heat. All right, so essentially, very fancy transformation. You transform high-resolution temperature data into high-resolution mortality predictions. The problem is that you have to validate it, right? Now we want to validate what happens in nature. So how do you do that? We will contrast the predictions against either ramping experiments, so you remain in the lab, right? You do measurements under constant conditions and you see whether you can predict what happens when you heat up the system, or you can do try to estimate what happens with mortality under natural or semi-natural conditions, okay? Let me show you what we've got. So for ramping experiments, we had information on the thermal death time curves for Drosophila, 13 different species, and these people were a group in Denmark. They did the characterization of TDT curves, and then they heated up these flies and obtained the critical limits, if you will, under 3 different ramping rates. And in here, what we have is the predictions of the model, right? Either the time that they would collapse or the temperature versus the results, versus what was measured. And you can see in this case, the R-square is borderline 1. So really predicting when they're dying, but also the temperature when they're collapsing is very near 1, so it's 0.96. So the model works very well. It predicts quite well what should be happening under ramping experiments. Okay. Now the other way to put this to a test was to contrast mortality in the field. So this is cumulative survival and the cumulative survival of this mortality here. Okay. It's the same thing. It's the third panel. Now, what we're contrasting here is the predictions of the model for warm acclimated and for cold acclimated against field abundance. Right. So Simon, you said you like demographic, so population. So here's demography. This is the population abundance measured in 7 different years empirically, and you can see that during spring, the population size of the Drosophila, the abundance would increase exponentially, and at a given time it would collapse over these 7 years. Right. So I don't know if you guys can see, but there's actually 7 different trajectories in here which are embedded in this, gray polygon. But you can see that when the population is collapsing, it coincides with our predictions of when cumulative survival, or you should have, relevant thermal mortality. Okay, so the model is predicting not only what things are happening in ramping experiments, but how population should collapse in the field based on temperature data. This is for Drosophila. We've also done some validation for fish. So in this case, we found this old review that I mentioned with nearly 300 thermal death time curves under constant conditions. So we got that review and we extracted these parameters for 52 species, and then we overlap that against the critical limits that was reported in another review published in 2000, and we could see an overlap of, I think it's 21 species, all right, from one review versus the other, and they had different conditions of thermal acclimation and heating rates and so forth, all right? We were able to control for that statistically, so at the end we could, essentially predict 141 trials over these 21 species, sorry. And again, here's what we predict, the temperature we predict, the time that we predict versus the observed, and you get R-squares that are, pretty decent, 82% in one case, or 0.99% for time. So the predictions also hold across species for fish. In this case, we had we saw this in the literature. We measured the thermal death time curves for Dichorogrammarus in the lab, and another group had reported mortality following 2 weeks of acclimation at these different temperatures. So we got their empirical data, which are the gray dots, and we ran our model. And this, again, these animals should be, dying at a given time above 20 degrees. And the model predicts very well now their, mortality following these 2 weeks of thermal acclimation. So again, this for aquatic amphipods seems to work as well. And finally, we had this capture-recapture experiment in Chile, and we went to the river. And so we got these freshwater crabs. We put 20 of them in these urine vials, and we opened like some holes so water could circulate. And we put these 20 individuals in this type of box. And in each box, we put a data logger. And we had this experiment where we had 5 of these replicates that were in waters that were exposed to the sun, and 5 of these boxes in waters that were under the shade, covered. And this is the temperature profile that we got for these different boxes. So these are the areas exposed. This is the areas that are covered. And for each replicate, we can estimate relative mortality, right? Because my students, they had to go there, every 2 days, and they would get the boxes, open the vials, see whether the crabs were dead or alive, and put them back. So it's a capture-recapture experiment. And what we see here is, here's who survived and who died. So this is a relative mortality, relative surviving here. And the data points are the empirical data, and the dotted lines are the predictions of the model. So again, we can predict not, in the 2 different conditions, survival based on lab data, based on lab measurements. Okay. So the validation across different organisms in different systems within species, across species, so far so good. We seem to be predicting quite well what's happening in terms of a validation. So what we've been proposing for a while now, and it's been a struggle, is a paradigm shift where we should be shifting for phenomenological descriptors of thermal tolerance, right? And static endpoints, essentially, that critical temperature that has low resolution, very low predictive power, and which comes historically from this ecophysiological perspective that we have something that we can measure and we can compare against, a species against another to see which ones are warm adapted versus cold adapted and so forth. That's fine. That works for that purpose. And so in that case, for instance, we're contrasting how organisms from different environments, for instance, how ecology explains physiological variation. Right? A tropical organism tends to exhibit a higher thermal tolerance than a temperate. That's fine. But the problem is that with climate change and global warming, we need now to adopt, better tools and close the loop. We have to adopt mechanistic models that explain better. They have to be dynamic because temperature varies in time. They have to be probabilistic because it's not 0 or 1, right? And if you have a population, you actually have like a fraction of the population is going to be affected by temperature. These models, ideally, they have very high resolution and very high predictive power. And the loop is when we're able to use now physiological knowledge obtained in the lab to predict what should happen in ecology, what should happen in their habitats and so forth. Okay, that's the idea of adopting the shift, or essentially going through this paradigm shift. So what can we do with this? And there are many things that we can do, and I'm happy to talk about some of the research that we've already done regarding these different things that can be done. So, for example, we can now estimate thermal selection in space and time, or across space and time. So in this case, we have this Litorina snail that was measured. They came from some Portuguese populations. We measured their thermal lifetime curves in the lab in the UK. And then we had some colleagues from Porto University. And these guys, I don't know if you know that they had like this science perspective a couple of years ago, where they put data loggers in 4 different places in each rocky shore, now in the Atlantic. So we have more than 1,000 data loggers deployed over the Atlantic in Europe, from Europe to Africa and in America. And they provide me their data sets, and it collects hourly temperature data, okay? And they provide me the data sets for these different places here, and we do simulations just as an exercise to see when temperatures will be restrictive for this particular Litorina. And you can see a few things here. For instance, the gray dots here is the data loggers under the shade, in the shade, and then the bright ones are data loggers exposed to the sun. And first thing, nobody dies when they're covered. So essentially, these mortalities here is organisms that are exposed to the sun. Second thing, you can definitely see the seasonal patterns in temperature, summer and winter, but you can see also that mortality increase, it also exhibits seasonal pattern. And the third, you can see This is micro variation between shaded and sunny in the same place, but you also may have meso variation in a spatial scale, right? So in one beach, for instance, you might have a lot of mortality or predicted mortality, whereas in another, because it's a more recluded city or it has more wind, if you will, basically no mortality whatsoever. So you could start, seeing how much temperature varies not only in small scales, but across, like multiple scales, and whether it has some refuges, for instance, and how this would affect things like population dynamics and the conservation of these species. Another possibility is that you can contrast or assess vulnerability across lineages within a given community. So we've done this review. Most of the data was published by Whitepack and his group in the British Antarctic Survey. And this is how the data looks like. This is the raw data. And essentially we use we like to call it statistical hocus pocus. We did some statistical analysis to retrieve what would be the shape of the thermal tolerance curve based in many instances on, just single data points. We were able to do it. I mean, there is of course some degree of uncertainty, but it's feasible. And from the thermal lifetime curves that we obtained from this data set here, we contrasted it against the temperature, in this case in the intertidal communities. These were data loggers that were deployed in the intertidal of Antarctica, and we simulated with the dynamic protocol, we simulated mortality of these different organisms, and you can see that a few of them in this particular thermal environment seem to be already dying depending on the temperatures that are reached in Antarctica during summer. And so you can see, for instance, in this case, arthropods and chordates, for this particular thermal profile, they exhibit higher mortality rates than others, than the other groups. And so we start trying to assess vulnerability based on thermal tolerance data. Interestingly, we also have the data loggers deployed 2 meters underneath the surface, so a subtidal, and then it's flat, right? I mean, it's very constant. And then the groups, they change, they inverse. Some of them seem to be adapted to high thermal variation, and the others by the opposite. We can now simulate the impact of heat waves. So there you go, Harrison, right? So this is a paper by Andrew Villeneuve. I like to say, just as an anecdote, this guy's the cousin of Denis Villeneuve, the director of Dune. And I'm not kidding. I asked him, he was like, yeah, this is my cousin. Like, wow. All right. But essentially what they did is they combined the thermal lifetime curve, right, which is here. They simulated these heat waves, and heat waves that were defined based on the definition of climatology, right? Some amount of temperature above, the normal and so forth. But they were arguing, which I agree, that the biological impact of that definition varies dramatically depending when the heat wave occurs. So for instance, in this case, this is the normal profile of the temperatures that they, it's all simulated, but this would be the normal temperature profile of that environment. And then they simulate the heat waves in different parts of it. This is summer, this is winter. And what they show is that for the very same heat wave, based on the climatological definition, you might have, let's go with winter first here. You don't have any impact in survival whatsoever, because it's a heat wave that occurred during winter, it's not a big deal. But when it occurs during summer, you have like this massive mortality that's being simulated with dynamic protocol. So you can study also the impact of heat waves with this approach. With some people in Netherlands, we're doing long-term predictions. So we measured the thermal lifetime curves now for these amphipods, the Dichorogamaros, that we validated with the acclimation data that I showed you. And what we did was we obtained this in the short term, and then with the computer simulations, we simulated what would happen following a whole year of different thermal profiles. So we would estimate the annual survival of these populations. And then we would plot the annual survival mortality, right, the predicted one, against a proxy of summer temperature. I don't remember now, maybe the hottest month of the year, something like this. And with a logistic regression, you can see that there is definitely, it's the summer, the most restrictive part of it that would determine annual mortality. And then with the climate forecast, the only thing we would do is, all right, so when would this river in the Netherlands exhibit this temperature? And we would just, instead of having the logistic regression with temperature, we transform the temperature into climate forecast, and then you have some idea of what might happen in the long future. So this is until 2100, so what, 75 years from now, how the system might be behaving based on the simulations that we have right now. Geographic distribution. So we've done by now, we have 2 papers, one in what happens with distribution limits based on thermal tolerance for freshwater fish. And my PhD student, André Molina, he's just published in Ecology Letters, showing what is predicted also for marine distributions. Right? Why? Because before, as you see in the literature, people were reporting these thermal safety margins, right? And what we did was we corrected now the thermal safety margins by the amount of time of the measurement. And what we saw is that the safety margins reduced dramatically. And then we can start predicting what might happen nowadays, because we know some fish are already threatened by current water temperatures. And we know that many of them are actually moving, right? And going to deeper waters or going to high latitudes. And so we can definitely use now this framework to study geographic distribution as well. And also the impact of multiple stressors. I've been talking about only temperature, but definitely organisms are being challenged by multiple dimensions. And in this case, what we did, this is the same freshwater crab that we validated in the study, remember, with capture data. So we measured the TBT curves for these three species, so freshwater crab, for these catfish and this gambusia, which I've heard that it's also an invasive species here. And we measured their thermal tolerance in normoxia, or saturated oxygen availability in the water, or in hypoxia, which was 50% saturation, more or less. And what you can see is that the thermal tolerance, it decreases, right? The amount of time that you can tolerate a given temperature decreases due to the lack of oxygen. And in the three species, you can see that there is a shifting in the slope, but actually, let's say the projection here that when you have a very high thermal stress, they essentially converge. Okay, you can now use this with some, again, statistical hocus pocus to combine the effects of temperature and the effects of hypoxia together in a single model. Okay, and you get these volumes, I don't know, these areas where you have now the mean temperature for a given day of the water stream or the lake. So here it is. And you have the oxygen concentration, and you can obtain for this, in this and here, what we're actually plotting is the survival probability. So the gray area is where the fresh crabs would tolerate this environment. In the blue one is where the catfish would tolerate, and then the most tolerant, which would be the gambusia. This is plotted in this pink or orange area. So now you can actually, try to start inferring what happens with thermal tolerance in combination with other stressors. A very interesting thing, this is coming now in ecography, hopefully in a few months. Very interesting thing, we have this in tabular form. So we provide a table that we want to give it to conservation agencies, and people could go to a river and measure the temperature or obtain the average temperature in that day for that particular location, and measure the dissolved oxygen and know whether it's being stressful for the organisms based on what we know from their thermal tolerance measured in the lab. And it could be done for the whole community if we have enough resources and time. And finally, community responses. I mean, many people have asked me about this. I've never done it, but it's definitely feasible, right? Where we can combine climate forecasts with this physiological data here, and then overlap it against different types of ecological networks. Could be pollination, could be food webs, and see how temperature might impact and cascade over the whole food web. So that's feasible. The problem with that is that, of course, it relies on measurements that should, in principle, be done across all possible nodes, which is a lot of work. And just to wrap up, as I said, most of my research has been focusing on animals, but then I was invited here a few years ago, started collaborating with people in Canberra and Sydney, and we realized that this is also generalized or generalizable for plants, right? And especially when they're measuring photosynthetic efficiency, and this is how it looks like. You can actually do this combination of different exposure times, so they expose the plants now to a thermal stress for 5, 10, up to 30 minutes, and different temperatures, and you can actually reconstruct the whole surface, right? The 3D surface here, you calculate LT50 or something that's a standard amount of damage, which in this case is 50%, and when you plot this threshold against stress duration, the log of stress duration, you get exactly what you'd observe, or what I've been reporting for animals. Okay, so it seems that in plants it works quite well. And since it does, of course, we ran the simulation of the dynamic model to see what would happen with leaf temperature now that was collected in the field. So this leaf temperature, I don't remember right now if it's in Canberra, but then you have these three TDT curves obtained from the beginning, middle, and end of summer. You can see the thermal tolerance decreasing as summer goes by, and we can now estimate how many times during these different this period these thresholds will be attained, and when you have like thermal stress based on our simulations, and you can see here that the maximum daily temperature, or in this case the maximum daily leaf temperature, starts decreasing as time goes by in the summer. And some of my colleagues are saying, so when you have, for instance, plants that have been desiccating throughout the summer, and then you might have something like a big heat wave that could induce a huge mortality. So they're also in the same problem that we have, right? So how do you combine in this case water availability with temperature and start predicting, especially the impact of heat waves? So to conclude, I would argue that thermal tolerance landscapes and thermal death time curves should be adopted as the common currency. Nowadays, after 10 years, I would be confident enough to say that actually the landscapes describe thermal tolerance. That's the trait. And T-crate is actually a proxy for the real trait. All right. So that's the common currency that should be used to study thermal limits. The dynamic models that we've developed, they provide more refined predictions, right, of what might happen in nature, and they improve overall predictive power at multiple scales. It could be, I don't know, you've seen, right? And finally, one of the big problems, but I think by now I can show you that I've showed that it's possible, is to validate the predictions in the field. Right? There are many different ways to do it. You should be a little bit creative, but it's definitely possible. But they remain challenging and they remain highly necessary. And the idea is this, right? We're right now stuck in here with some predictions that have very low resolution, and we need to change very fast to the adoption of this type of framework in order to increase the resolution of our predictions and hopefully do something about it. And with that, I'm happy to answer questions. Thank you very much.
I'll start off. Is thermal tolerance the right measure for predictive models? Given that we know that performance drops off well before a thermal maximum is reached. And the wonderful thing about thermal tolerance, and I've done many of them, is that it's easy. It's a really easy measure, given all the complications on protocols and how you measure it. But it worries me that we put, for predictive models that we want to use, whether taking the easy approach is the right approach.
Point taken. Yeah. We in Sydney now with plants, we had exactly the same discussion. Can I go back to it? Okay, yeah. So one thing that we've been discussing in Sydney is because most of the mortality in plants is in principle associated with this desiccation or lack of water availability rather than thermal tolerance per se. And then it's like, well, let's forget about thermal tolerance, let's go into water, right? But conceptually, what's actually happening is that no matter what kind of stressor or what kind of challenge you might be studying, it still falls in this two-dimensional axis, because every organism is exposed to temperature, and whatever happens to them happens in time. So it's still, I would say, enlightening to understand whatever kind of problem, even if it's a different trait, right? If it's a different challenge, it's still enlightening to understand what's happening in this frame. For instance, with a discussion with these colleagues, they would say, well, it's water availability. No problem. What's actually probably happening is that water availability is decreasing the thermal tolerance of these plants to such an extent that at a given time, when they experience that particular temperature, they all die. So then we say it's not the temperature per se. No, sure. It's the combined effect. But whatever mortality detect of a given organism, it'll be detected within this bivariate space. Right? I think the big mistake that we do is sometimes just think that actually it's temperature, the major driver in the absence of everything else. So we're assuming that we're stuck here in the best world, in the best case scenario where there's no other challenge, and the only thing that really matters is temperature. And what might be happening is that these combined effects ultimately, resulting in death or high mortality rates, but we're missing that additional component, which is the detriment, if you want, of thermal tolerance. So you have like physiological, damage before the thermal effect per se.
So are you putting a safety factor in there? Where you can put a 10% or 20% safety factor in, because there are many departments now wanting to be able to put that kind of safety measure in.
I mean, explicitly no. Right, and actually, part of the reasons why I got into the area, coming from this evolutionary physiology stuff and more empirical, not theoretical, was that I had problems buying the idea of a thermal safety margin if you cannot explain how they evolve, right? Because suddenly you say, oh, these organisms can tolerate 25 degrees higher than what they're experiencing. I'll buy it, but explain me how did that tolerance or that safety margin evolve in the first place, right? So I think there are two problems in there that are, occurring at the same time. One is the time component, because they're doing measurements that are very short time. So that means that you're really, tolerating something that's very high, but in the long term, that's not sustainable. So there's a time component, just an experimental artifact. But the second one might be the fact that actually many organisms, they're evolving under these constrained combined stressors. And so when you measure them under benign conditions, you think that they have a safety factor which might not be there. Right? But I'm very skeptical of the safety margin as a concept if there's no mechanistic explanation of how they emerge in the first place. And for instance, one thing that we've been working now with corals is that corals shows us, right, as a thermometer, that by the time you have these heat waves or you have a challenge and a fraction of the community is dying, right, that they're right in the evolution of a safety factor. Maybe those that remain alive, they come back and the coral community reconstructs itself or regenerates, and then in principle that community should have a higher safety margin than before the selection event. But then you can explain by in terms of evolution.
That was a great talk. Thank you very much. Okay, so the relationship you've shown is extremely compelling, and what I'm trying to resolve in my head is how does that sit with sort of the model that you will have different physiological responses to acute events versus chronic? So how do you explain this sort of continuity that you observe when there are probably different things going on against different stressors?
Absolutely. I mean, when I started working on this, I had this very, I would say, naive, suggestion here that it's inverted. Let's go here. I don't know what's easier for people, if it's temperature in the x-axis, y in the x-axis, but anyway, all right. Let's imagine that you have something like this. I would argue, or I started arguing, that those individuals that had a very high intercept and a very steep slope, so they would lose their thermal tolerance very rapidly in time, they were adapted to acute conditions. Right? Because that means they can tolerate, a small amount or very brief bursts of thermal stress, but it could not be prolonged, in the long term. Nowadays, with the dynamic model, you can actually test that with simulations. Right? So it's possible. You can do that with simulation. Is there a trade-off? I proposed in 2014, and not many people like the idea. I don't know. When you plot the intercept versus the slope, you can see that they're correlated. We saw that for animals on the one hand, and now people with plants, they're observing the same thing. So it might be that you have some organisms that are capable of tolerating an increasing mean temperatures, and others that are more prone to tolerate acute changes. It's something that we've seen like with the Antarctic data set, and I mentioned, right? So you have like for the intertidal, it impacts a set of organisms, and when you go to the subtidal, it impacts the opposite. But I don't know exactly if you had like a jack of all trades, it would be ideal. But it's something that I presume could be modeled easily.
Maybe building a little bit from that question, you mentioned that the thermal tolerance landscape should be the measure of thermal tolerance.
Mm-hmm.
But then there's some thermal selection experiments where they select for one measure and then they don't get a separate measure of thermal tolerance. So they get a response to selection in whatever they selected for, but another thermal tolerance trait doesn't get pulled along with it. And then people would be saying, well, maybe these are two different traits. I mean, within this framework, they're both the same, they're both measures of thermal tolerance. But yeah, I guess it's across that landscape, there's actually probably many different thermal tolerance traits, which each have a different physiological mechanism of tolerance, and each probably have a different set of genes to, And how does then the evolution of a thermal tolerance landscape look?
There's many answers to the question. So the first one of the big problems that were very critical about arguing that there were different thermal tolerance traits was the fact that, they would measure different proxies, and these proxies were not correlated. And then they would say, oh, it's different genetic traits, or, under different genetic mechanisms or whatever. And we said, like, it could be simply lack of statistical power, low statistical power. And you're inferring something about, genetic control just because you don't know how the trait looks like in reality. right? And by the time we had the landscapes, there are many possibilities. For example, you could, assuming that the slope and the intercept are not correlated, you could definitely increase or change just the slope and have the same intercept, and you could have a curve here. So you could be selecting for, let's say, tolerance for, chronic stress without any changing here. And even though, and if you measure, resistance or, in short-term experiments versus long-term experiments, they wouldn't be correlated, but it's still the same trait. This could happen, and we've seen it actually with acclimation experiments and all. We've seen it in different systems, and so one of the things that we did when we ran the dynamic model, when we ran this, was showing by the time we predict, for example, this. So people, they were measuring these CT max under different ramping conditions, and they weren't correlated. So they say, oh, if it's a slow ramping, it's one trait. If it's a fast ramping, it's a different trait. We said, no, we predict all of them. We can predict all of them. And it's just a problem of statistical power in that case, right? I mean, definitely you can have, different traits. But coming back to it, there's a very interesting thing. You talk about different genetic controls or different physiological bases. And this is anecdotal, but I find it very interesting. I was watching the 2012 Olympics, and I was watching Usain Bolt running the 100-meter sprint. And I was working on this because it was in the functional ecology in 2014. And then I said, look, if we're talking about metabolic intensity, which is temperature, right? You increase the temperature and animals start, using energy faster thermodynamically, right? Maybe I can find in the athleticism records, in the race records, the same response that I find with thermal tolerance. So I got to like the world records of athletics or whatever, and I downloaded all the records from 100-meter sprint up to Ironman, which was 100 kilometers, not even marathon, 100 kilometers. I could show you the data, but and I plotted the average velocity, which is metabolic intensity, just like temperature. So the average velocity of how that race was won against the record time in log scale. So I mimicked this here, right, with temperature and it was a log perfect, man. It was 0.99, and the only 2 points that got out of the curve were the 100-meter sprint and the 200, because you have anaerobic metabolism. And so the interesting thing, what caught my attention from that particular thing is we were talking about different athletes with different shapes, right? I mean, different morphology. And you can invoke like different genetic control, whatever you want. It was still following the 9-9 line, So I would say that the TDT curve, it is how I call it, an emergent trait that encompasses to some degree, with some degree of precision, this interaction between different biological and physiological processes. And even the exception to the rule in the athletic, when you go into anaerobic and you actually go out of the, right, because it's the switch between aerobic and anaerobic, you go out of that curve for the Olympic records, is also a good model of how you should interpret what might happen with thermal tolerance trait, that yes, you might have sometimes a few traits or some physiological response that might change dramatically how you respond to that thermal challenge. For example, activation. And just like another parenthesis, you guys have to think about the curve or the thermal tolerance as a labile trait. You acclimate, so it's not a fixed trait, right? It's like physiology, it's like metabolism. It can go up and down depending on your thermal history. So even though like most of the noise that we're seeing right now is because of ignoring time, but the trait itself, the TDT curve, it will go up and down in time as well. And we know it changes throughout development, and it should change throughout, with different thermal regimes, like acclimation temperatures as well. So it's not a fixed trait, it's more, it's flexible.
Unfortunately, we're out of time. So I'm sorry. Enrico will be around all week though. So if you have more questions, do you mind if I blow up your spot and tell people they can reach out to you? But yeah, thank you all for attending. I'm sorry for the technical difficulties. Next time I'll reserve the room for an hour before so I can get things set up. But yeah, thank you so much for being here.